Today we're going to start general and special senses. Now let's talk about the eye. The eye is one of the most complex organs, and one of the most special senses is vision. Remember, know the eyelids and that they're associated with the exocrine glands. Know the structures associated with production and secretion and removal of tears. And know that the extrinsic eye muscles. We're going to speak about all those next. The eyelids, they also have another name called palpebrae. There's the upper and lower eyelids join at what we call the medial canthus and the lateral canthus. The medial canthus has the glands that secrete gritty sleep found in the lacriminal caruncle. Basically, if you ever woke up with sleep in your eye. They also have sebaceous glands, and they can become infected, and that's known as a sty in your eye. Next is an area called the conjunctiva, or conjunctivitis. There's irrigation or damage to the conjunctivas called conjunctivitis or pink eye. The lacriminal apparatus includes some main things: the lacriminal gland, the lacriminal canals, and the nasolacriminal duct. Remember that the lacriminal apparatus produces essential tears, and it distributes them across the eye and removes them. The lacriminal gland secretes the tears and is superior and lateral to the eyeball. The tears drain through two pores at the medial canthus called the lacriminal canals and the nasolacriminal duct. This is an image showing you the lacriminal gland, the lacriminal pores, and then the nasolacriminal duct. Let's talk about the eyeball. The vascular layer of the eyeball contains blood and lymphatic vessels and the intrinsic eye muscles. The functions include providing a route for the vessels supplying the tissue, adjusting the amount of light entering the eye, providing a route for secreting and reabsorbing the aqueous humor, and then controlling the shape of the lens. The vascular eyeball has some structures we need to know about. Number one is the iris. The iris has the pupillary muscles that change the size of the pupil, the window to the eye, the ciliary body, which contains the ciliary muscle and the ciliary processes, and the suspensory ligaments, which adjust the shape of the lens for focusing. Know this picture of the eye. You see the fibrous layer, the vascular layer, and the inner retinal layer. We're going to dissect the sheep eye, and here you're able to see the cornea, the sclera is the white of the eye, the iris, the ciliary body, and the choroid. You're also able to see the posterior cavity and the anterior cavity. Know this image. You will have to label this in your test. This is showing a dilated pupil and a constricted pupil. The pupillary dilator muscles extend radially away from the edge of the pupil, and that results in an enlarged pupil. On the right, we see the pupillary constrictor muscles form a series of muscles in concentric circles around the pupil. When these sphincter muscles contract, the diameter of the pupil decreases. We have two sets of receptors on the eye called rods and cones. The rods are used in dim light and found on the periphery of the retinal surface. The cones are used in bright light and detect color. They're found in the macula, and that's the center of which is the fovea, or fovea centralis. You might have heard of something called macular degeneration. There are some chambers of the eye. There's in the anterior cavity and the posterior cavity. Problems with the fluid and pressure is a condition called glaucoma. That's in the posterior cavity. That posterior cavity is filled with vitreous fluid in the vitreous body, and it holds the retina in place. We'll see that when we do the dissection. This shows you the anterior cavity and the posterior cavity. Now, the lens, it's held in place by the suspensory ligaments, and it changes shape to accommodate for focus. This shows you three things. Know this, when the focal distance is closer than the light source, the longer the focal distance. And it says the rounder the lens, the shorter the focal distance. So this shows you when the focal distance is short, long, or just right. Again, this shows you on the actual eyeball, and you can see the rounded lens and the flattened lens, and you can see the focal point on the fovea of the retina. Let's move on to two types of situations that happen in human vision. There's something called nearsightedness, called myopia. Make sure you know that. Nearsightedness is myopia. And if you look closely, if the eyeball is too deep or the resting curvature of the lens is too great, the image of the distant object is projected in front of the retina. So in myopia or nearsightedness, the image is projected in front of the retina. Now let's go to hyperopia. That's farsightedness. If the eyeball is too shallow or the lens is too flat, hyperopia results that the muscles contract to focus, and the image is projected beyond the retina. Now, remember that we talked about photoreceptors, and what they do is they respond to photons. Photons are units of visible light, so color is determined by wavelengths. Photons of a red have the longest wavelength, where photons of violets have the shortest wavelength. The longest wavelength has the least energy, where the shortest wavelength has the most energy. Now let's go back to the rods and cones. The rods respond to the presence or absence of photons regardless of the wavelength. The cones are different in that they respond to three types of wavelengths, or three types of cones: blue cones, green cones, and red cones. There's something called color blindness. Color blindness occurs when one or more types of the cones are not functioning or missing. The most common is red-green color blindness, where the red cones are missing. It's more common in males, about 10% more than females. The total color blindness is extremely rare, and it's one person in every 300,000. The structure of the photoreceptors: there's an outer segment that contains hundreds to thousands of flattened discs. They're made of something called rhodopsin, and that contains opsin and retinol, and it's derived from vitamin A. Retinol is the same in rods and cones, and opsin is different. Finally, let's go on to the anatomy of the ear, and we'll finish up. Know this: there's the external ear, the middle ear, that's where that amplifies the sound waves, and the internal ear, and that contains the sensory organs for equilibrium. Make sure you know the parts for matching. Know the auricle, know the auditory ossicles, know the oval window, know the bony labyrinth, know the cochlea, know the auditory tube, know the facial nerve, know the temporal bone, know the round window, and know the tympanic membrane. The external ear is made up of the auricle, or the pinna, and it's a fleshy cup, and it directs the sound into the ear. The tympanic membrane is the eardrum. Make sure you know that. It's a thin sheet that vibrates when sound waves strike it. The middle ear: there's an area called the tympanic cavity, and it's an air-filled chamber. There's also an area called the eustachian tube, or the pharyngeotympanic tube. There are three small bones called the auditory ossicles. Make sure you know them. They're in the middle ear, and they connect the tympanic membrane to the internal ear. There's the malleus that attaches to the eardrum, the incus that attaches to the malleus and the innermost bone, and the stapes. And it has the base to nearly fills the oval window into the internal ear. Let's talk about hearing. Vibrations of sound waves determine a stimulus. The tympanic membrane vibrates the ossicles. The pressure pulses and travels through the perilymph of the cochlea. The pitch or frequency is determined by which part of the cochlear duct is stimulated, and the volume is the intensity, and it's determined by how many hairs are activated at that site. Lastly, the 6 steps of hearing. Once the sound waves strike the tympanic membrane vibrates the auditory ossicles. The vibration of the stapes applies pressure to the perilymph. The pressure distorts the basilar membrane. The movement of the basilar membrane distorts the hair cells against the tectoral membrane, altering the neurotransmitter release. And 6, the impulse travels through the central nervous system through nerve number 8.